that today. There's green, PowerPoint. Good. All right, so again, reminder, exam here is Wednesday, right? Wednesday it'll open all day to take it. No lab Wednesday, no class Wednesday. So Wednesday's a study day for immuno. Tomorrow, for those, all of us at Preston are in micro, right? Micro exam day tomorrow, opens tomorrow morning. Give you all day, take it. We will have lab in micro. We will do Haemophilus tomorrow. Today in lab for immuno, it is dry today, so it's the case study review day. So we can connect some of the dots through all the chapters that we have. For this one, we'll have a case study for each chapter, I think, for each. And then today we're finishing up chapter 12, which is the testing, molecular testing. I did bring a couple of PCR examples to kind of read through to let you know kind of how the procedure works for that. And that's what's going on over the ABI that we're doing real time, not real time. I keep seeing real time. It's reverse transcriptase polymerase chain reaction. Okay, so we are going to point that out because, as you say, get used to saying something like RT-PCR. Okay, you hear it. I've heard it real time said before. So it kind of sticks with you, but I don't want y'all to look at it that way. So that's where we are. We're going to finish up our molecular testing. So we're over on the FISH, right? FISH, which is a hybridization method. So solution hybridizing FISH technique. FISH, you'll hear that. I've heard it for years, you know, referred to testing that they use. And so we'll go through this and how FISH works. So we really, so it is not an array method, right? It is a hybridization. I guess you call it a hybridization array. So we're on, you got any questions before we get going? Do we have, how many do we have in? We got 4 on Zoom. We got 7, 10, 13. So got everybody. Okay. All right.
What about lab midterm for immuno?
Lab midterm. What am I doing?
When is it?
Like,
Oh, when is it? I think it's on the schedule for next week, right?
Yeah.
and will we have like a guide or anything? A quiz?
Not without lab on Wednesday. No, his income's right back on Monday, right?
Yeah.
It's on Monday. Yeah. Yeah, we'll talk about it, but it's just covering what we've done so far. So. Yeah, don't sweat too much, but you've got make sure you got all your stuff back. But we've done a serial dilution, we've done dilutions, math problems, and we've done ELISA. I think that's all we've done, right? And then we won't have the new lab will be the electrophoresis, which we'll do next week. Well, I say that we're gonna probably be playing with making it up and just see. Because I forgot about the midterm being sitting in there. What's that?
With bone dilutions on the exam, how bad was that a straight through left grade?
If you don't do them right,
Yeah.
it gets counted off. Yeah.
Well, I mean, yeah, but like how much is it, like what's the percentage of the midterm?
Like if you
for-
don't-
Oh, I don't
Well,
know.
what
Say
is
there's
long?
20 questions, you might have 6 or 7 dilutions to work out. Kind of things that gonna give you an idea. If I remember right. So yes, you will be tested on whether you know how to do dilutions. You won't get a partner to help you out. So if you rely on a partner to help you out, first go for all those 3 sets. You better be working on those. I promised you I was going to make sure you knew how to do dilutions. All right? Okay. Okay. But yes, the midterm will cover what we've done so far. So, and dilutions are a big part of that. ELISA is a big part of that. And agglutination beginning of agglutination reactions with the titer, antibody titer that we did. So every lab that we've done so far will be covered in the midterm. So it can usually go pretty good, but it can go really bad too. So, and it is, and it's right before midterm grades are posted, so you'll have a 1 hour, not a very good grade in immunolab. It doesn't work out very well because it's a big part of your grade, the midterm. What was that?
Is the antibody titer the little pellet thing that we did?
Yeah, that never really played out. Yeah. Yeah. You won't be doing it, but you will be asked to see if you know what's going on. You're not going to be sitting at the desk doing anything for the midterm. You'll be taking a test.
Well, we need to know the numbers like 4+, 1+, that type of stuff, or.
Not on this one, no. I don't think I have you grading it, no. You just where it's positive last and what that means for the titer. But you have to figure the titer. You gotta tell me where it is. So yeah, I can't remember how many questions it was. And we've added ELISA from the last time we did midterms. But at this point last year, they were already doing some agglutination reactions. So they were doing some RPRs and stuff like that. So we hadn't quite got there yet. We're waiting on those to when we cover it in the lecture itself. So I'm trying to build everything as the lecture goes. That's why the electrophoresis has been moved up, because we're in that part now. And then when we get into the autoimmunes, that's when we'll pick up the tests. So I think immunolab's going to get better as far as, hey, we're testing something and there it is. You're just screening for loops. All right, let's go over FISH. Okay, so this is fluorescence in situ hybridization. So hybridization group. So a probe, what they're doing is, and what FISH lets you do is actually use the cells. So across this test wells are actual tissue cells. Okay, so you're looking for something inside the cell with FISH. Okay, it utilizes a nucleic acid probe. So hopefully that's familiar with you since y'all been studying, to identify the DNA. So we're looking for DNA inside the cell. The target DNA is found inside an intact cell. So that's the difference with FISH. We're using intact cells, and we're using probes from a few thousand to hundreds of thousands base long. So the bases that we talked about on Friday, the bases that we talked about, those are you can see that you're adding to a probe has a length of bases, and it's going into the cell. It actually enters the cell, and it binds to the DNA that it's looking for, that probe will. And what you see is color change, okay? So we use reference range. Here you have a test DNA plus the reference DNA, and then you can see what cells have, the test, because it would only be test by itself, which would leave it at green. If we found the DNA in the cell that we're looking for, green and red make yellow, and you can see that the normal equals the color ratio of 1, yellow and orange. Gains in amplification grow to green, and then losses in amplifications go to red. So this is how we would go about reading and looking for if there was something wrong with the cell, okay? That's the green one. That means that we have something, the cell's doing more than normal, okay? So out of control, if you want to put it that way. So it's a good way to look for abnormal cells, right? So where do we use fluorescent in situ hybridization? We use fluorescence, but there's also a kind of a similar test known as CISH. I guess we'd say CISH, C-I-S-H, and that's using just chemoluminescence labeling instead of fluorescence labeling, and that allows you to use a regular microscope instead of a fluorescent microscope, and that's the only difference. So anytime you see ISH, that would be an in situ hybridization. So what are we looking for? It says, where was that? We're looking for identifying differences in chromosomal abnormalities. And y'all, I don't know if y'all remember that first video, to save a life video, when the kid had some, you know, in abnormal cells, he had cancer, and it was a genetic translocation. Y'all remember that video that I showed during orientation? Yeah, it meant a lot, right? It was very moving. Probably moved you to tears, you know, seeing this kid, you know. No? Y'all don't remember that? Well, part of that, you know, we look at chromosomal differences, chromosomal abnormalities are detected with fish. Okay, so we can also use them for T-cell lymphomas. We can also use them for B-cell malignancies and graft versus host disease after transplantation. So it's a good way of picking up, and it's an old way. You'll hear people using, hey, we use fish, the hybridization method, to find the abnormal cells in the patient. We were able to diagnose the patient because we were able to locate the abnormal cells. Okay, so it's a way of identifying that. We also have a bead array. And I think this one, where is that one? Bead array. Figure 1214. Get the bead array up there at the top there. For nucleic acid analysis, bead array antibodies are replaced with single-stranded oligonucleotide complementaries to test nucleic acid. And we get a bead array in the method here, I think. Let's see. Where did I remember reading about bead array? I think it was, yes. It was before we talked. It was the array method. The slide kind of out of place. Did I go the right way? No, I didn't, did I? Did I? Yeah, I'm going the right way. That just seems out of place. I don't know. Because it came before fish and fish came after the, anyway. So bead arrays are kind of what we use today. We kind of use this bead array detection, multiplex detection of proteins and nucleic acids. So we use it for tissue typing or respiratory virus panels. The beads may have antibodies or single-stranded oligonucleotides attached. So basically it's like a detection. So you mix in your sample. If the sample has a specific link or specific DNA, then the bead array will detect it. So we're all about detecting with hybridization method. Oh, here we are. So if you're using the antibody, the beads have specific antibodies to the actual DNA of what you're looking for. So it will attach. And when it attaches, it has a labeled antibody that attaches and it shows up. So it kind of, you know, you see it. Arrays with oligonucleotides. Here, you just have a specific oligonucleotide. It sees the sample DNA and it attaches to its complementary band. So once that attachment happens, then you see the amplification, or the signal is then detected. So it's all about, we're searching DNA, we're searching RNA, and we found these different ways, but we're doing what? We're adding segments of what we're looking for. Okay, so we're making complementary strands. So the important part of COVID was to do what? Get its genetic code, to get its DNA or RNA for it. It was an RNA virus, but to find out what those base sequences were for the COVID-19. That way, the testing then gets used. So when we talk about PCR here in just a minute, the whole point was once we found out, and it didn't take long. Once they, you know, found the virus, it wasn't like this virus was hiding. They were able to, you know, collect the virus. They were actually able to find the RNA, sequence it, see that it was new. That's where the novel name came from. It was a sequenced virus that we hadn't seen, and then we could start testing for it. Okay, which meant we would take a sample of the virus. We have the technique that we'll go over with. We break it open, and then we have all these little segments of DNA chasing that RNA, and then attaching to it and finding it. So the whole point of a positive test is you have that unique sequence of RNA from this virus. So that's why it's very, if you want to say, it's positive, okay? Because you're not going to have that sequence otherwise. So it was just kind of how we had to catch up, but this was still the go-to test. It just took long, takes a long time. And we'll go over the length it takes. This is from your book too. This is on 179. This is solution hybridization. You see we have the test RNA with a biotinylated capture probe. So here's a probe. So if we wanted to say this was our COVID test RNA, and then we would put a capture probe, okay, and this one's using streptavidin coded support. So it basically binds that, okay, and this little capture probe here then links on to the coded support, and then we can send the antibodies in or capture antibody here. We can do different ways of capturing it, but the whole idea, and this is a gold electrode, so we see some of that time, we'll see gold being used. But variations of the method to detect specific DNA and RNA, probe hybridization in the solution, which is we take a viral mix, that swab that you have is put into a viral transport media, whether that's saline or something different, and that was part of the issue with COVID was we didn't have enough of that. So they'd have these swabs and like, we don't have any transport medium. And everybody was having to scramble to find a acceptable transport medium because the companies couldn't produce enough. So then somebody came up with just saline would work. So they were putting the swabs in a tube of saline, sterile saline, and sending it. Well, as long as it was captured that way, they were able to test it. So all these techniques, are definitely, the basic, what I want you to know is taking the test, whether it's DNA and RNA that we're looking for, and then trying to capture that by using known sequences to capture the DNA or RNA, and then we have to have some way of signaling us that we got it. All right. Oh, here's more about fish, sorry. Somehow, why did fish come? Where did this slide jump in here? I was thinking we were already there. Anyway, review then for fluorescent in situ hybridization. That's what FISH stands for. Then sections of solid tissue deposit of cells on a glass slide. Probes are covalently attached to fluorescent dye. Okay, and the technique can identify chromosome abnormalities, which we've already talked about. Now this is what we want to talk about, PCR. This is where we're going to spend a majority of our time today. So target amplification, this is, we take that, this is what we're looking for, right? This is we have a template of DNA to attach to what we're looking for, and we mix that all together. So polymerase chain reaction, basically on the polymerase chain reaction is you add everything that you need for the process to go forward. Okay, and it's a long list. Let me go over that. So the procedure results in an amplification of a target DNA or an RNA, in our case with COVID, okay? So we're gonna add everything we need to do to amplify that sequence. So if you need it, you add oligonucleotide primers, okay, that prime the synthesis of the copy. DNA polymerase is added, okay, to join the deoxynucleotides. You add a buffer with a pH that makes it optimal for this procedure to take place for the polymerase activity. And these oligonucleotide primers are the key components of the specificity of the PCR reaction. So we have to know that. So the primers are created, okay? So how do we get those primers? Well, we would get positive samples, right? If we get positive samples of the actual COVID, if we want to take what we're doing here over in ABI, that if we have those, then we can denature those and use those to detect if the patient has. Okay, so it's a real so here are the we were sent these because we were going to help. We were planning on helping them do this testing this summer, and basically we've been put on like standby. So here is the extraction part, and here is the reverse transcriptase PCR setup. So we have two different ways they're working, or both are working out over their ABI. So we have a nasal swab. Short-term storage is up to 48 hours at refrigeration. Okay, so they can bring them in, put them in a refrigerator for how many days? 72 hours, 24, 48, 72, 3 days, all right? If we got a long-term storage, we're gonna put those at minus 80 degrees Celsius. So we're gonna put them in the freezer. The viral RNA extraction protocol is by Qiagen Viral RNA Mini Kit. So you gotta have a kit for this to do this RNA extraction. So the reagent prep is you add buffer to lyophilize the carrier RNA, and the aliquot calculate the volume of buffer based on the RNA mix needed to batch extraction. And what we do is we add the N is then multiplied by 0.56 mils to yield Y mLs. Okay, so we have to do some math where N is the number of samples to be processed simultaneously. And most of these come in a wet, like a little, some of you use, the HNID plate. Kind of looks like that, but it's not that deep. It's a little thinner well, a little small. And we have a little pipetter, that has like 8 different tips on it at the same time. So you're going to do that, kind of do a lot of pipetting. So N's the number of samples to be processed, and most of these little things have about 36 samples, well, 36 wells, but that only allows you so many samples. I got y'all really confused yet? Little? Okay. Well, Y is the calculated volume of buffer. Z is the volume of carrier RNA buffer. Gently mix invert tubes 10 times to avoid foaming and do not vortex. Okay, so to do this, the buffer carrier RNA mix is prepared fresh by adding ethanol, 96 to 100 proof. Did y'all know we had that on campus? We had like 100 proof alcohol on campus. Y'all didn't know that. All right, so then wear your PPEs, and viral RNA extractions are performed in the biosafety cabinet only. So they don't want us taking these samples. They've actually closed like a staircase over at ABI. It's only for transport from the ground up and then back down. So there's a staircase that's designed for sample transport. Like you can't take it through the lobby. You have to take it up the staircase, and it's in boxes and stuff like that. So what do we do once we get it all extracted out? It's a whole procedure where we add the vortex, apply centrifugation up to a minute, repeat steps 7 to 8, entire sample run through the column, add the buffer, centrifuge again for a minute, replace the collection, add 500 microliters of buffer, centrifuge full speed for 3 minutes, replace the collection tube, spin for a full minute. What are we doing right now? We are extracting out of the virus. So we have real samples, and we have viruses in those samples. If they're there, we're having to what? We're having to denature that virus to expose what? The RNA that we're going to test. So we have to destroy the virus. We have to open it up. So all these procedures are to get the sample ready to be tested. Okay, so that's the RNA extraction part. The next part is the reverse transcriptase PCR setup. We have aliquots of IDT N1, N2, RP primer probes. So we take those, we're going to have on our plate, we're going to have the probe, right? We're going to mix that, all the aliquots, put the enzyme mix on ice, keep the RNA samples on ice, wear your PPE. and it's performed in the hood or the thermal or next to the thermocycler. So we got this thing called thermocycler. It's going to read the plate. So N again is the number of patients that we're going to have, and it's 3 plus the number of samples. And then N equals N plus 1 for samples 1 through 14, and N equals N plus 2 for samples 15 or greater. So we have to get all this up together. Mix each master mix well, pipetting up and down using the 96 well plate. Place 15 microliters of each master mix in the appropriate well. Add 5 microliters of nuclease-free water. Add 5 microliters of extracted RNA, to the appropriate N1, N2 RP wells. Place optical sealer on one end of the 96 well plate. Roll across the top to avoid wrinkles and bubbles. Seal the edges of the optical sealer. Spin the 96 well plate for 3 minutes. And then place the 96 plate well in the Bio-Rad thermocycler, load and run. So if you haven't read this chapter already, with real-time with reverse transcriptase, PCR, oh, that's gonna get me every time. We're having to do what? We are having to have computer help. Okay, so that thermocycler is going to go through, and what we're going to do is it's going to be in like at least 2 times. All of this is going to go through the thermocycler. Let me show you where I'm reading this. For many procedures, we pre-mix the PCR, primer, and buffer, and apply them to manufacturers which template DNA. And you see that you've got this 3-step PCR cycle. So I'm on 179. A denaturing step, we did that one right. An annealing step, so the temp pass, and then the extension step. So what we're trying to do is apply the probe to the actual RNA that's from the virus. Okay, so that we got to give it time to do that. So it's, think of your ELISA, giving it time to find, if it's there, find the sequence that it's searching for. And then we're going to let that do its business, kind of let it start doing what? Replicating the RNA, okay? So that's going on. If it's there, it's going to go a little bit longer, so it's going to be able to see. And then you have a computer that's going to detect whether it's there in the well or not. Okay, so let's take this. We got our PCR, amplification of the labeled primary is incorporated. So here you can see, but I kind of want to do here, no amplification, no label incorporated. So what you'll see is the computer is going to read the electrogram here, and what you'll see is if it's there, if it went ahead and amplified this section, then it will peak and show that it was there. Okay, so you have this kind of baseline. So every well plate you do has to have a positive, known positive in there, because you're going to compare, the computer is going to compare that well to the patient wells. Are y'all kind of sort of understand this procedure?
So if you have one well that doesn't test positive, you can redo the whole thing?
No, you would just call that patient negative. Oh, if your sample doesn't show positive, yes. So you could have to redo the whole shooting match, yes. If you've got a control on there that has to read positive, you've got a positive known sample, and if it doesn't read positive, then the whole whole plate is invalid. So yes, that's part of the issue as to the length of time that it could. So if it doesn't work, right? Well, guess what they have to wait on? They have to wait on known positive standards from the health department. So a lot of the delay in our testing up here, getting it going, was getting known positives. Another delay in the testing up here was that the procedure was lab created, which meant that we were using the CDC's protocol, and it really didn't match what they wanted to do with this manufacturer that was giving them the stuff. So they had to work on validation. So yeah, it's not an easy thing. And a lot of the testing, if you saw, a lot of the testing was approved by emergency release with the FDA, which meant that the FDA was allowing some testing to go through before it really had all the information that it wanted for this testing. Because people, if anybody tweaked a procedure, like I'm going to tweak it here, I'm going to do it this way, I'm going to change that sample or that collection. Every time you do that, you have to ask for another emergency authorization from the FDA. So it's a mess, okay? It's been a mess. But we're kind of getting control of the mess, but it's now October, and this has been a mess since March. Okay, so if you understand, that's what we've been dealing with, okay? Some testing has been better than others. Some has been very, very good. And some has had issues, okay? All right, so what we're trying to do is that we can do transcription-based amplification, okay, and that's kind of what we're doing here. So let's look, we got the RNA here. If we want to run this one like, hey, we've got this RNA, just like if this was the COVID RNA, we're going to reverse transcriptase it. So we're going to add reverse transcriptase. A tail primer comes in. Okay, so this is the tail primer. And what you're going to see is the complementary DNA strands start to build. Okay, so you add RNase 2, that gives us a single strand of that DNA. We add a second primer, reverse transcriptase. We get DNA starting to form up. And then at the end of that, we get multi-copies of the RNA that we started with. So if you know what you're starting with, doing this procedure gives you a resulting RNA amount that can be detected, okay? So I don't want to get into every step of this. We don't want you in the, I mean, we want you to, at the end of your career or in your career, to know how to follow this and maybe help out. Some of you might go into the industry of molecular testing, and sure, you would need to know this whole thing. But if you don't understand what transcription is, then you don't understand what's going on here, okay? So this RNA, if you were going forward from the DNA, then this is what you would end up with. So what we're doing is taking the virus's RNA and going backwards. So we're reversing the steps. We're going from RNA back to DNA and then going forward again once we get the DNA there to give us the new RNA. Does that make sense at all? Hopefully y'all can follow that. All right. So another amplification. So all of these amplifications are to basically what? End up whatever's detected in small amounts. So if you think about one virus's RNA, that's a very, very small amount of RNA. But can we take that small amount and amplify it to the point of we can detect it? Find some way of detecting the virus, whether it's HIV, whether it's hepatitis, or whether it's COVID. Can we detect this virus, very small amount, and so we're amplifying that little bit that we've found and amplifying it to an amount that we can say, yes, it was there, okay? Ligase reaction. Ligases are going to break the sequences so that they can be amplified that way. So they're going to split them up, break them up. So here's a strand displacement amplification. So we have ligase chain reaction, probe amplification. We have strand displacement amplification. And then we actually have base DNA amplification. All right. I think what I want you to do is stay like focused on the PCR description, the definition, okay? I think that'll help us get through this part. really good thing that I see on the 183 is this real-time quantitative PCR signal generation. And you see that you get so many more copies, and that's like 10 to the 7th, 10 to the 6th, 10 to the 5th, 10 to the 4th. So what you're doing is you have a threshold, and this is how the computer reads with PCR. The samples have to be above the threshold. So when you do the graph on the computer, you're going to come up off 0, and you're going to have a noise line, and that's known as the threshold. Okay, that doesn't pick up, it picks up all the wells, what's in the well, but then all of a sudden you'll have it go beyond the threshold, and that's positive. That's how the computer picks up the positive in the well. So that's important to know, is this threshold. And that's where you take your known stuff. You take your negative samples and all the noise that's in there, set your 0. It's kind of like using a spectrophotometer and setting it at 0 with a blank. You're using, you've got the water, you've got the cuvette, all of that will absorb some of the light in the spectrophotometer. When we talk about testing with light, well, here's the same kind of way. All those wells have some RNA in them or DNA from the sample. You got other stuff in there. So there has to be this jump above the threshold of the noise that's down here at the basement. And once that jumps, then it will detect that as a positive sample. Because you can, when we looked at it the other day, when we were over there, we opened up the computer program, and part of the testing was to establish the threshold. That each, once all this testing gets set up and they run knowns and they verify it and they verify their procedure, that was to all give you a baseline threshold. And that was set in the computer. And you set that up and you put a new well in there. But what it does is that when you have the positive well, the computer is going to look for that to increase just like it was before. It's going to see that increase in the positive well. And that's going to be compared to all the other wells, especially your patient, right? Okay. Needless to say, this is why it took, why you don't get your results back. It takes you a long time to run this. It takes you, you're limited by how many wells you have. You're limited by the time that it takes to extract it. And then you're limited by the thermal cycler, how many times it has to go through before then you have to verify your results and then report them out. So it's 4 to 5 hours of work just to get the result back. And then you're limited where you might have, like at the beginning of the outbreak of COVID, we had, 10, 20, 30 samples being collected. And then all of a sudden now they're doing, thousands a day. And then you bring that into the lab and go, okay, we just got a box of, 500 samples. When were you going to get those done? Yes.
How does the 15-minute test work?
The 15-minute test is totally antigen based. So this is looking for RNA. We got to extract out the virus, actual viral RNA, and amplify it to detect it. The antigen test is using an antibody to the antigen of the virus. So its coat has antigens. You see it with a coronavirus, you see crowns around it. All those are antigens. So they take that known antigen now, develop a monoclonal antibody, impregnate a piece of filter paper that's going to let it run across just like we would a flu test. And then it binds it and lights it up if it's found. Okay, so that's the 15-minute version of detecting antigen versus all of this to determine an RNA sequence.
So if your 15 minutes comes back positive, would they then do this, or do they do this with everybody?
Actually, what they're doing is they're calling, if you're antigen positive, you're positive. They're not going any farther. If you're antigen negative, they're doing this. The antigen negative is a presumptive negative, just like the strep negative on a strep test, throat swab. Then we're doing another step. So the next step would be to send that sample to be reverse transcriptase RNA detection, PCR. So the problem with the long delay is that you get, with me, I got exposed on a weekend. I got notified that I have a exposure. So I knew Thursday night I go get tested. I don't know my result until over a week from the time I got exposed. So how many people supposedly had I exposed in that time period? So to stop the spread, the whole idea was to find that person who's positive and get them quarantined before they can pass it on. So it was all timing, and the timing was stinking, it was really stinky. Because first off, I was not contacted by anybody to tell me about the positive test. I was never contacted about my positive test. So everything is kind of foggy with this testing method, the isolation. So the only good thing I can say is I still see you guys wearing masks here, and we're wearing shields here. The bad thing is I don't see masks being worn when you're not here. Just, that's up to you. But I'm seeing more and more laxing of the mask out, which, as you know, some states have actually let their mask mandates expire to this point. So it's individual groups and individual places that are still asking for the mask to be worn, but it's kind of a, and has it helped? Has the mask helped? Who knows, right? Numbers aren't really moving downward, but they're not really shooting upward either. So they're just kind of, as they say, flat right now. 500 to 700 to 800 cases a day. But the antigen test is driving that too. That quick turnaround you're seeing, quick testing.
So if your antigen is negative, but this comes back positive, would you be considered a false negative? Or
Oh yeah, with the antigen test, yeah. Yeah, and that's the problem with the antigen test. It does not catch it sometimes. That you miss it with that test, but you capture it with this test. Are y'all's head spinning right now or not? Good. The good thing about this is we can put it to a real light, and we're not having to just guess as to why these tests. But when all this testing came out, when we were in the middle of virology, which you'll be in next semester, and we basically played this was our lab project as we went through. It was just this idea of what is a PCR test, and how does it work? Probably should show y'all a video of that. I think we have one, don't we? I think we do. Yeah, I think that's the way I'm gonna finish lecture today. All right, I'm gonna stop sharing this screen, and we're gonna move over so you can, I'll let you watch one of our PCR testers. Zoom is still here, I take it, yes. Okay, so remember I told you we have a YouTube page for A-State Micro. I think I can find it, maybe. There it is. I have one subscriber, so not very many followers. Okay. Let's see, we got, how do we want to do this? We got 3 minutes. Which one? Yeah, I'll let y'all vote. We got, Kelsey's doing a Snapchat, so I don't know. That was only 50 seconds. I want to eliminate Kelsey's. We have treatment. We really don't want to do treatment. We can do either. I think this one will be a good one. 3 minutes long.
Okay, you
I'll pick it. Oh, hold on. I got to share the screen. Okay, share the screen again. Okay. Okay, this is Emily Marshall. She's going to give you COVID testing. So this is kind of a preview. Y'all be doing something similar to this next semester.
So, my name is Emily. This is my presentation on the COVID-19 testing process. The novel coronavirus, or COVID-19, is the name of the disease caused by the severe acute respiratory syndrome coronavirus 2, known in short as SARS-CoV-2. It is a viral test and an antibody test for COVID-19. The viral test is for current infection and uses a real-time reverse transcriptase polymerase chain reaction. The antibody test may be ordered online or through a healthcare provider and is used to test for past infection by detecting IgG and IgM antibodies. The RT-PCR test is the more commonly used test right now because it diagnoses acute infection when the disease is still contagious. The real-time reverse transcriptase polymerase chain reaction, or RT-PCR, is used to test for the SARS-CoV-2 virus RNA in upper or lower respiratory tract specimens, including nasal, nasopharyngeal, oropharyngeal swabs, sputum, lower respiratory tract aspirates, bronchoalveolar lavage, and nasal aspirates. It is a qualitative test, so it will have a positive or negative result. RT-PCR is used to make copies of short sections of DNA from small samples so that it can be tested and detected. This process is called amplification. SARS-CoV-2 is an RNA virus, so it needs to be made into DNA before it can be amplified. During the test, reverse transcriptase binds to the virus's RNA and makes a DNA-based copy so that it can be amplified. The process of amplification is achieved through repeating heating and cooling the sample in cycles. When this happens, the DNA is denatured and separates into single strands. The intact polymerase is used to synthesize two or double-stranded copies of the DNA using the original as a template. If a sufficient amount of viral RNA is present in the sample, it will be amplified and detected, and the test will be positive. If no viral RNA is present, the test will be negative. In a single-plex format, the test uses 3' controlled sets to detect 3 regions of the SARS-CoV-2 nuclear capsid gene, and 1' probe set to detect human RNase P. When multiplexed into a single reaction, the test uses 2' probe sets to detect 2 regions of the SARS-CoV-2 nuclear capsid gene, and 1' probe set to detect human RNase P. I found my information for this presentation from Blackboard, which is under the Food and Drug Administration, Lab Tests Online, the World Health Organization, the National Human Genome Research Institute, and the Centers for Disease Control and Prevention.
All right. Okay. Y'all got any questions? Stop the share. Questions from Zoom? Oops, don't want to play that. So, Zoom questions, nobody questions? All right, see you downstairs for dry lab, case studies, immunology. Got White for those helpful.